All right, we talked about what microevolution is. We talked about the different type of variation, quantitative or qualitative, that you can find in natural population, and which one are genetics or environmental, how you can figure that out. And finally, we talked about some population genetics, and in particular, we focused on the Hardy and Weinberg principle and equation, which allows you to figure out what the frequencies of the genotypes and the alleles should be in a population if this population is at equilibrium, so if it's not undergoing evolution. So what we're going to talk about now instead is what are those factors, those agents that ultimately cause microevolution in a population, so that either introduce new variants or they change the frequency of the existing variants. And then finally we'll talk about how are these variants preserved in the population, even though sometimes they might be lethal or greatly disadvantageous. So how are they preserved? Why do we need to preserve these variations first of all, and how this happens? So let's start with the agents of microevolution. So these are all these different events that we're talking about that cause microevolution. Not surprisingly, all of these, if you look carefully later on, are all violations of the Harding-Weinberg conditions for genetic equilibrium, which you remember we talked about earlier on, or in the first part of this lecture, which are basically that there are no mutations, so no new variants can be generated, the population is close to migration, it's infinite in size, there is random mating, and so on. So you see later on, all of them are violation to these conditions that were put by Harding-Weinberg as necessary for a population to be in equilibrium. So, what are these agents of microevolution? So there are several of them. First one, mutations. By mutation, what we mean, and we talked about that in BIO358, are any changes that take place on the DNA. So we have changes that could be deletions of a nucleotide, substitution of a nucleotide, they can be in frame or so on. Different mutations. Mutations, changes in the DNA can introduce new variants, as you can imagine. Variants that can be completely neutral. They might not cause anything. They can be deleterious or lethal for the organism that has them, or beneficial. So now, when it comes to those rare beneficial mutations, so that they introduce some beneficial variation in some locus in a population, now natural selection will be helping them. Obviously, helping them between brackets. Obviously, that will be somehow selecting these adaptable beneficial variations and make them more frequent within the population. So mutations for sure is the first agent of microevolution, which can introduce additional variants, which then can, if beneficial, can actually spread throughout the population. Other agent of microevolution is gene flow. By what we mean by gene flow is the introduction of new genetic variants, which is caused by flow of gene, basically by mixing different populations. And this is caused by two important factors. First of all, you need to have movement of organisms or their gametes. For example, with plants, you can have pollen going from one side to another one of a mountain, right? It can mix two populations with their genetic pools. So there must be movement of these organisms, and also these material that's moved, whether it's the full organisms or the gametes, must reproduce. Obviously, must be spreading its gene, therefore changing the frequencies of the variants in its own genetic pool. So obviously, this is a violation of the Harding-Weinberg condition of closed population. So now you have influence, there is migration of either new organism or their gametes, and these obviously change the frequencies of the alleles. One example you have here is a blue jay, just carrying acorns from a tree to another location is already spreading the genes from these plants and the population where the plants belong to into a different population. So common thing. Now this is different from genetic drift. Oftentimes, students confuse gene flow and genetic drift. Genetic drift, instead, it's all based on chance events, events that are not really caused by migration of populations. They're caused by migration, but basically they're caused by events that lead to the change of the population or the formation of a brand new population, not mixing in this case. So these chance events are totally unpredictable. They violate the Harding-Weinberg condition of infinite population size, and ultimately usually they reduce the variability within the population. So they cause microevolution by reducing, eliminating some variants and changing obviously the frequencies of the other, but overall reducing the genetic variability. There are two main mechanisms of genetic drift, which are called population bottlenecks and founder effect. So the population bottleneck is that effect that you see when a population gets greatly, drastically reduced in size suddenly, whatever is the reason. It could be a natural disaster or man hunting. This will reduce the population size. Imagine you're going from 1 million to a few hundred individuals. This has a huge effect on the frequency of the alleles. Most likely will result in the elimination on subvariants, okay, random elimination. And now you have that small population that has been reduced in size, if it's allowed to repopulate the earth, like, will form a new population which has dramatically different frequencies on the alleles compared to the original one that was subject to the population bottleneck. So one example you have here is what happened with the northern elephant seals. These are seals that are present mostly on the west coast of North America. They were greatly hunted in the late 1800s, and as such they were basically extinct. There were only 20 survivors left at a certain point. Then hunting was banned, and these 20 survivors could actually give origin to the thousands of northern elephant seals that are present nowadays. But as you can imagine, that event of hunting, so population bottleneck, has greatly reduced the variability within these populations, so making them more susceptible to extinction even in the future. Second type of genetic drift is the founder effect. This takes place when you have few individuals from one population detaching from one population and establishing themselves elsewhere. By doing that, so if you take a small group of people from a large population, it's similar to the bottleneck effect. If you think you're taking these people that don't necessarily have the same gene pool than the overall population, even though they belong to that population, but just by chance you might have different distribution in genes in that group, small group, and you separate them, you create a new population. This new population will be, in terms of variation, different from the original one, and therefore evolution, microevolution would have taken place. One example is what happened in the population in Charleroi and Saguenay-Lac-Saint-Jean, which are regions in northern Quebec, a few hours, 3 hours from Quebec City. If you keep going up the St. Lawrence River, you'll hit those areas. In this population, for example, which was established by a few hundred people, the incidence of the disease, myotonic dystrophy. This is a disease that basically is characterized particularly by muscle weakness. It's a dominant disorder. It's extremely frequent in this population, much more frequently, 10 to 100 times more frequent than in the general population in Canada. So the reason being that these individuals that moved to this area originally, these few individuals, had a higher incidence. They were not reflecting the gene pool of the population they were coming from. They were chosen just by chance. Unfortunately, they had a higher incidence, which they carried over to the whole population that is now obviously has populated this area. Obviously, there are big conservation implications of genetic drift, whether we talk about bottleneck effect or the founder effect. And obviously is that by reducing the genetic variability of this individual, particularly when it comes to the bottleneck effect, you're now recreating a population which has very little variability. And variability is necessary for survival. That's what natural selection works on. So if you have less variability, your chance of survival in the long run is much lower than the one of the original population. For example, you have the cheetahs in Africa that 10,000 years ago they were greatly reduced by an environmental disaster. Even nowadays, 10,000 years later, they still have an extremely reduced genetic variability. So they're still greatly susceptible to potential extinction. So these effects are felt for a long term. So bottleneck particularly is a very disastrous, if you want, event in terms of potential extinction of a species. Other agent of microevolution is what is called the non-random mating. So one of the big principles of Harding-Weinberg equilibrium is that all individuals are random mates. So the chance of mixing different gametes is the same for all the gametes. This is not really true. We all do. We all choose a mate. For example, we don't really take anybody as our own mate, as us humans, right? If you think you are looking for certain characteristics, it might be different from individual to individual, but we all have certain characteristics in mind in terms of aspect of look, obviously personality too, and look as well. These obviously are greatly affected by commercials, by newspapers, and so on, images that are given to us. That's the reason why many of them, for not myself, and many of us spend more time at the gym compared to the library. So there is a selection there at that point, and this obviously, by not mixing randomly, favors certain type of traits, variants within the population, which obviously it's a direct type of example. One example here, or specific aspect, is what is called sexual selection. Sexual selection is a subtype of natural selection, which is based on exaggerated phenotype, for certain type of exaggerated phenotypes. This was already hypothesized by Darwin, but basically this is what you observe in different animals, where you have sexual dimorphism, where you see that males, for example, that start having some enhanced structures in order to attract mate. It's exactly the consequence of non-random mating. There's certain things that attract the female, and therefore the male, between brackets, adapts and undergoes sexual selection, pushing these dysmorphic traits, like a long tail, for example, in this bird. You can see these birds here. If you have a shorter tail, you have much lower chance, to mate compared if you have a longer tail, right? So that is dimorphism favored by natural selection. This type of character and enrich the population in this type of character, therefore causes microevolution. So here is a summary of all the agents of microevolution we just mentioned. So mutations, gene flow, genetic drift, non-random mating, and the last one, which I already mentioned when we talked about Darwin, is obviously natural selection, which, if you recall, is that process in which some variants, which we call heritable traits, adaptive traits, beneficial traits, become more common in a population. And the reason being that these traits give an advantage both in terms of survival and replication to the individual that carry that, right? So this obviously is a violation of Harding-Weinberg principle, in this case of infinite growth, and this acts on the phenotype, doesn't act really directly on the alleles, but on the phenotype. So if you have that specific phenotype, whether you have only the allele or you're heterozygous and so on, you have an advantage, which again, I want to stress, that's one thing we always miss in terms of natural selection, is that these traits has to give an advantage in terms of survival, but also in terms of replication. And this can be quantified measuring what is called the relative fitness. Relative fitness, which you see here on the bottom, is actually a numerical value which tells you the number of surviving offspring that an individual with a specific trait has compared with the general population. So for example, if you have an adaptive trait that makes you more resistant at low oxygen level, and you can have 2 offspring compared to 1, the relative fitness will be 2 divided by 1, so 2. So that's what really gives an idea of whether these or how natural selection is working. So natural selection can work in different ways in terms of effect. So it can work on quantitative um, um, variation, like the one we mentioned before, for example, height, weight, that they distribute in discrete increments. So there are not only 1, 2, or 3 variants. Here you have so many variants that distribute in small increments, giving origin to this bell. So now, natural selection can act on this type of variation and therefore change these type of curves in different ways. So it can act by directional selection, stabilizing selection, and disruptive selection. What do we mean by that? Let's look at the different effects. So when we talk about directional selection, what we mean is that natural selection is actually changing the distribution of the variance for a specific character by changing the curve towards one of the 2 extremes, so either the higher extreme or the lower extreme. This is an example. So for example, here it's acting on these birds, pushing the length of the tail. So if this is the distribution of the length of the tail, this is moved towards, so the mean value is moved towards a higher level. So the bell has been moved to the right side. Okay. One example is with predatory fish when they are some guppies, for example, they're increasing in overall size. So if this was the distribution of the size originally, now they're getting larger and larger. And the reason being that the predators, they tend to eat the smaller ones. Okay, by doing that, they're selecting progressively those guppies that do have the genes that makes them bigger, and therefore the population is enriching in these larger variants. So that's called directional selection. Second type is stabilizing selection. In this case, the distribution of the variance, instead of being pushed towards one side or the other, it's actually pushed more or extended towards the top. So stays the same, but more gets compressed toward the mean value. This is the most common mode through which natural selection acts. There are several examples we can use with that, but the classical example is what is happening in human newborns. Like it's kind of reverting nowadays, but throughout history, the size of the newborn babies has become more and more and more stabilized towards a mean value here of around 3 kilograms. So the bell was a little bit larger here, but now it's become more and more towards this value. The reason being that the mortality at lower weights at higher weights is much higher than at this value. The reason being that at lower weights, the babies or the humans carrying the genes that make them be born small would die, because in the old days, if you were born at 6 months, good luck, there would be no way to keep the baby alive. Instead, if the baby was too big, most likely would not have been able to come out, right? And therefore will cause the death of the mother and as well the baby inside. Nowadays, these things are changing, which is actually a problem. The reason being that now with the modern techniques, with the NICU, for example, we can now keep babies alive even at 5.5 months, 6 months, right? Newborn baby, like extremely premature, can still survive. The same thing for large kids. With a C-section allows now even large kids to be born. And so these genes, these variants for large babies are spreading more and more and increasing in frequency in the human population. The problem is that the body, the female body, has not adapted to that, right? So this is fine in modern countries where you have the technology to keep the young baby alive, the premature baby alive, or to do the C-section, but in other countries this can be a big problem, as you can imagine. So the last type of natural selection is called disruptive selection. And this, instead of pushing towards one side or increasing, if you want, the incidence of individual with a mean value, in this case actually divides basically the bell into two separate. So it pushes the both extreme. So basically lowering the amount of individual towards the mean value here. So it pushes basically the extreme phenotypes there. One example is what happened to the Galapagos finches bill size, which they were distributed, so or they are still distributed in this way, but in case of extreme drought, actually the extreme drought is causing natural selection, which somehow gives an advantage to birds that have either a smaller and longer bill or extremely large bill, and is lethal to the one that have an average size bill, and therefore causing this disruptive effect on the distribution of the bill size in these finches. So it just summarizes the three mechanisms. Directional selections basically pushes the value, the variance, towards one or the other extreme. Stabilizing selection instead increases the frequencies of the variance which are close to the mean value, and disruptive selection instead increases the frequency, if you want, of the extreme variance, reducing the one close to the mean value. Finally, let's talk a little bit of how all the variation is preserved in living organisms and why it's preserved. Variation is essential for living organism, right? The reason being that natural selection thrive only if there is variation. It can work only if there is variation. One example is the bottleneck effect, right? That we were mentioned before. The reduced variation really puts individual at high risk of extinction. The other reason why it's important to have it, and one of the reasons it's important to have, is because obviously there might be some alleles that can make you less fit to survive in the current environmental condition. But environmental conditions keep changing. It's possible that what is not advantageous nowadays, it might be advantageous in 1,000,000 years. So you need to keep variation within population so that you can have adaptation in several directions instead of only one direction. So how is this achieved? Well, there are different mechanisms that preserve the polymorphism. That's what we mean. We use the word that we use to describe the presence of lots of variation, polymorphism. So one of them is diploidy. Now, most organisms are diploid, or they have more than one copy of chromosome, and this is a great way to protect recessive alleles. Why? Because even if they're lethal, right? If they're lethal means that if you are homozygous recessive, you would die, for example, right? But now, if being recessive in heterozygous, that phenotype is not expressed, and therefore individuals that are heterozygous can still survive, and they can keep the allele alive. Obviously, it will be extremely low, the distribution of the allele in the population, but still is kept there for eventual success in natural selection if conditions change. So diploidy is one mechanism that protects, if you want, polymorphism of recessive alleles. Another example is balanced polymorphism. And we talk about balanced polymorphism when you have that not only one phenotype is protected or is selected by natural selection, but you can have two or more phenotypes that are maintained by natural selection in the population. One example is what happens with sickle cell anemia. So sickle cell anemia is caused by a single point mutation in the gene that codes for hemoglobin, for one of the subunit of hemoglobin, that changes basically one amino acid from hydrophilic to hydrophobic. The consequence of this point mutation and this amino acid mutation is that the red blood cells, they lose their donut shape, normal donut shape, and they get this sickle shape. right? Looks like a half a moon. The problem with that is that while the donut shape red blood cells that can easily move through the capillaries, the bloodstream, they're designed to move extremely well. Now, the sickle ones, they do not. So they have a tendency to actually clog small vessels, causing, you can imagine, like infarct, strokes, different type, obviously, of necrosis downstream, and so on. So, as a result, these individuals that have this variant here, if they are homozygous recessive, they would have sickle cell anemia, and they would have anemia, but they would have high risk of stroke, infarct, so cardiac arrest, and so on. However, individuals that are heterozygous, so they have obviously the normal version of hemoglobin, and they have the sickle version of hemoglobin as well. So some of their cells are actually sickle, but they're not extremely sickle. So they still preserve their capacity to move through the blood, so they have reduced chance of having heart attack or stroke and so on. At the same time, because of the shape of their cells, their red blood cells, they're actually less susceptible to infection by the parasite that causes malaria. So because of this, now the heterozygotes actually have an advantage in some situations. So if you are born in an area where malaria is very common, my mother, for example, she was heterozygous. She was born here in the north of Italy in an area where it was very swampy. Malaria was very common in the old days. And so for this reason, she was actually heterozygous, because by being a heterozygous and having these modified, the sickle type of red blood cells, she was actually immune to infection from malaria. So she was resistant to malaria. So as you can see now, you have pressure here, or an advantage of being both homozygous dominant if malaria is not present, or heterozygous if malaria is present. So, and this is exactly what we mean by balanced polymorphism. You have that now selection is maintaining the two phenotypes stable, the reason being that both of them provide some sort of advantage, whether malaria is present or not. Now, another type is that selection. So and this to talk about again how polymorphism is maintained, is that selection sometimes is different depending on the environment. So some variants may be advantageous in one environment, some other variation in another one. One example is the garden snails here. So you have all these different coloring of their shell, which give them different advantage depending on where they are located, right? So brown ones, and they have an advantage in areas rich in woods. The striped one have an advantage if you are in a grassy field, and so on, like this one, and so on. So it's the variation might be or the preservation of the variation might be simply due to the fact that the species is spread through many diverse environments, and therefore all different variants have an advantage compared to the other depending on the environment they find themselves. Another example or another mechanism through which polymorphism is present is through what is called frequency dependent selection. And this is basically a mechanism of selection which varies depending on the frequency of a specific phenotype. So what I mean is that you may have that a variant is advantageous when the frequency of that variance is very low, but is disadvantageous when the frequency is high, and vice versa for the other variants. So the fitness of that variant might change depending on its own frequency. One example is what happens between predator and prey interactions, where you have that, for example, you may have a variant that is not preferred. Let me say there's one variant that is preferred by a predator, right? And the predator, so when there is lots of that variance, the predator will go and get that variance, right? Once that goes low, the predator might start adapting to actually eat the alternative variant, which before was giving an advantage to that species. By doing that, adapting to the other variant, now you have that the original variant is called variant A, the one that was eaten originally, now start having an advantage in terms of survival. And then this lasts until variant B is greatly reduced, and then the predator may start readapting to variant A, and so on. And so you can have fluctuation in terms of frequency of the two variants, but both of them keep being preserved over time. And then in some case, well, the last thing I'm going to discuss here, you may have what happened neutral selections, where in reality there is no variant that really gives you an advantage or a disadvantage, and therefore all these variants are kept in a population. So in summary, finishing up both part 1 and part 2, what did we talk about in this lecture? Well, first of all, we define what microevolution is. So we talked about how microevolution is nothing else that change in the frequency of different basically allele within a population. Then we talked about what are the variations found in natural population. We talked about quantitative versus qualitative variation. Then we talked about what are genetics, and that's the ones that microselection works on. It's genetic variation, not the environmental one, and how you can figure out which variants, variability are genetic and which one are not. Then we talked about population genetics, the field of genetics that studied the frequency of the different alleles and genotypes in population. And in particular, we focus on the Harding-Weinberg principle, an equation that allows you to determine, in theory, what should happen to a population if microevolution is not taking place. What values, what frequency of a certain variable you would expect if there was no evolution, and therefore figure out whether evolution is happening or not in a population, in a study. Then we talked about what causes microevolution. We talked about mutation, gene flow, genetic drift, bottleneck effect as part of genetic drift, founder effect, the same thing, and then non-random mating, natural selection. And then we talked about how natural selection work by enriching a population of some extreme phenotypes or mean phenotypes and so on. So we talked about directional, stabilizing, disruptive type of natural selection. And finally, we finish up talking about the mechanism that are involved in the preservation of several variants in populations, so the polymorphism, preservation of polymorphism in different loci, which is necessary again for any species to survive. Any species needs to have lots of polymorphisms in themselves in order to be able to adapt to the ever-changing conditions in the environment.